Welcome back to the deep dive. Today, we're really tearing into a fascinating biological puzzle. It's one that just netted the 2025 Nobel Prize in Physiology or Medicine from Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi. They were recognized for figuring out something called peripheral immune tolerance. So our mission today is to unpack this. How does your body's own incredibly powerful immune system hunt down invaders, viruses, bacteria, things that are masters of disguise, yet somehow avoid attacking you? We're going deep on the immune system's own security guards, these regulatory T cells or Tregs, and the genetic switch, FOXP3, that basically tells them what to do.
Yeah, and what's really striking here is just how radical this whole idea was initially. For ages, the main theory in immunology was what we call central tolerance.
Okay, central tolerance, what did that involve?
The basic idea was that self-tolerance, telling self from non-self, it happened only in the thymus. That's sort of the T-cell boot camp. Any T-cell showing signs of attacking the body's own stuff, it was supposed to be eliminated right there early on. Deleted.
That sounds neat, but maybe too neat? How good was that system, really, if that was the only line of defense?
Well, that's the thing. It wasn't foolproof. Not even close. Our sources suggest the deletion efficiency, how well it worked, was maybe only 60, 70%.
Wait, 60 to 70? That means, what, 30 or 40% of potentially dangerous T cells were just getting out, leaking into the system?
Precisely. A pretty big chunk of cells capable of causing autoimmune problems were always escaping into the bloodstream. Which raises a huge question, right?
Yeah, how did we even survive as a species if the main factory checkpoint was that leaky? Sounds like a recipe for disaster unless there was, like, a backup plan.
Exactly. And that gap, that apparent flaw, is what Shimon Sakaguchi started questioning back in 1995. He proposed this idea of peripheral immune tolerance. Think of it as a second, active layer of control operating outside the thymus all the time.
Ah, okay, so not just relying on the initial training.
Right. His experiments, they were groundbreaking. They identified this specific group of T cells. Their whole job was to actively suppress these unwanted immune attacks. These were the Tregs, the regulatory T cells, the internal peacekeepers.
Okay, so Sakaguchi identified that what these peacekeeper cells exist, but the how, the genetics behind it, that came later.
Yes, that piece of the puzzle clicked into place around 2001, thanks to Mary Brunkow and Fred Ramsdell. They were studying these mice called scurfy mice.
Derby mice.
Yeah, they had these really severe, always fatal autoimmune conditions. Think massive inflammation, skin problems, organ failure, just uncontrolled immune attack. And Brunkow and Ramsdell asked the key question: what single genetic mistake could cause such total immune chaos?
And they found it, the master switch.
They did. A mutation in one single gene, FOXP3. Get this, just that one faulty gene caused the entire immune system to collapse in those mice. And the human connection was, well, tragically clear almost immediately.
How so?
Because mutations in the human version of FOXP3 cause this devastating condition called IPEX syndrome.
IPEX.
Right.
That stands for immune dysregulation, polyendocrinopathy, and neuropathy, X-linked. For listeners, what does that actually mean for a person?
It means total loss of immune control. The immune system starts attacking basically everything. You see severe gut inflammation, that's the enteropathy, attacks on hormone-producing glands, the polyendocrinopathy. It showed, without a doubt, that this one gene was absolutely critical for keeping the immune system in check.
And this is where Sakaguchi's work and the FOXP3 discovery came together, right? Which is always the cool part in science.
Absolutely. In 2003, Sakaguchi's group connected the dots. They proved FOXP3 wasn't just, hanging out on Tregs. It was the master regulator.
It actually made them Tregs.
Yes. They showed if you take a normal T cell and force it to express FOXP3, boom, it becomes a fully functional Treg capable of suppressing other cells. That really cemented FOXP3 as the key controller, dictating the development and the suppressive job of these Tregs, making sure those escaped self-reactive cells stayed quiet.
Okay, so we've got the origin story and the genetic command structure for these peacekeepers. But let's talk mechanics. When those potentially rogue T cells do escape the thymus, how does this peripheral tolerance system actually work? What are the layers?
Right. Peripheral tolerance is absolutely crucial because, like we said, those self-reactive cells are always slipping through. It's not just about preventing autoimmunity either. It also keeps your immune system calm about everyday things.
Like the food you eat or the bacteria in your gut.
Exactly like that. You don't want a massive immune flare-up every time you eat lunch. And you definitely need tolerance towards the trillions of helpful bacteria in your gut. In fact, there are specialized Tregs, called induced Tregs or iTregs, concentrated in the gut for precisely this reason.
Okay, so how does the body achieve this calm? What are the mechanisms?
There are several ways the body enforces tolerance out in the periphery. The first one is kind of passive. It's called ignorance.
Ignorance. Sounds a bit dismissive.
Well, it sort of is. Basically, a self-reactive T cell might recognize a self-antigen, something on your own tissues, but if the concentration of that antigen is super low, or if it's hitting behind physical barriers, the T cell just doesn't get a strong enough signal to activate. It remains ignorant.
So anatomical barriers play a role.
Oh, definitely. Think about places like your eyes or your brain, what we call immunoprivileged organs. They have extra physical barriers to limit immune cell access and activation, protecting those really sensitive tissues. Low signal or no access equals ignorance.
Okay, so if the signal is too weak or hidden, nothing happens. What if the T cell does get the signal, but something else is missing?
That brings us to the second mechanism, energy. This means functional unresponsiveness. Think of T cell activation needing two keys. Signal 1 is recognizing the antigen. That's the first key. But to really kick off a response, it needs a second key, signal 2, which comes from things called co-stimulatory molecules, usually on antigen-presenting cells. If a T cell gets signal 1 from a self-antigen, but doesn't get that crucial signal 2, it doesn't just fail to activate, it gets shut down. It becomes anergic.
So it recognizes the self-signal, but without the danger confirmation signal, it's told to stand down permanently.
Pretty much. It enters the state where it can't respond properly, even if it encounters the same antigen later with co-stimulation. It's a key safety check.
That makes sense. A two-factor authentication for immune attack.
You could think of it that way, yeah. Then there's quiescence. This is simpler. It's just the default state for naive T cells. They're basically parked, metabolically quiet, in a state called G0. They just circulate, waiting. They only wake up and start dividing if they get both the right antigen signal, signal 1, and the co-stimulation signal 2.
Okay, so ignorance, energy, quiescence. What's the last resort?
The final backstop is peripheral deletion. This is basically apoptosis, programmed cell death. If a self-reactive T cell somehow gets activated, despite the other checks, or maybe its activation is just too strong or persistent, the system can often trigger it to self-destruct, cleaning up the mistake.
It really is a multi-layered security system.
Yeah.
And it's not just the T cells managing themselves, right? Other cells are involved.
Absolutely. Dendritic cells, or DCs, are major players here too. They're like the scouts, constantly sampling the environment. Sometimes, particularly immature DCs, might pick up self-antigens from normal tissue turnover. When they present these to T cells in the lymph nodes, they often do it without providing strong co-stimulatory signals.
So they deliver signal 1 but withhold signal 2.
Exactly. That interaction basically tells the T cell, hey, this antigen is self, nothing to worry about. And that encounter typically pushes the T cell towards energy, or sometimes dilution, or even helps convert it into a regulatory T cell itself.
Okay, this is fascinating. But here's where things get really complex, almost contradictory, right? Because these Tregs, these essential peacekeepers preventing autoimmunity, they become the bad guys in cancer.
That paradox is really at the heart of so much modern immunology and drug development. Let's take autoimmunity first. In diseases like lupus, SLE, rheumatoid arthritis, RA, or juvenile idiopathic arthritis, JIA, the core problem is a breakdown of tolerance. Tregs are often there, sometimes even increased at the site of inflammation.
But they're not working properly.
Well, two things can happen. One, the inflammatory environment itself, maybe it's flooded with signals like TNF-alpha, can make the attacking T cells, the effector cells, resistant to Treg suppression. It's like the Tregs are hitting the brakes, but the effector cells just ignore them, their brakes are shot.
Okay, resistance. What's the other problem?
The other issue is that the Tregs themselves can become unstable in that highly inflammatory soup. They can actually lose their FOXP3 expression, stop being suppressive, and sometimes even start producing inflammatory signals themselves, like IL-17. We call these unstable cells ex-FOXP3 cells.
Wow, so they can actually switch sides.
In a way, yes. They contribute to the inflammation, particularly seen in things like JIA, joint inflammation.
So for autoimmunity, the therapeutic goal seems clear, if difficult. You need more effective Tregs. How do researchers try to achieve that? Boost the peacekeepers?
One key strategy hinges on a specific protein. Tregs express heavily the high-affinity interleukin-2 receptor, also known as CD25. Think of CD25 as a super-sensitive antenna for IL-2, which is a growth factor for T cells. Tregs have these high-gain antennas naturally.
So they can pick up even tiny amounts of this IL-2 signal.
Precisely. Much better than other T cells. So if you administer very low doses of IL-2, it preferentially boosts the existing Tregs, making them multiply and function better without overstimulating the other potentially dangerous T cells.
Ah, clever. Using their own biology against the disease.
Exactly. And this low-dose IL-2 approach has actually shown some real promise in clinical trials for diseases like SLE and even alopecia areata, which is an autoimmune hair loss condition.
Okay, now let's completely flip the script. Cancer. Here, Tregs are the enemy.
Absolutely the enemy. We talk about tumor-infiltrating regulatory T cells, or TI-Tregs. They accumulate in huge numbers inside many solid tumors, sometimes making up like 30, 40, even 50% of the CD4 plus T cells there. And generally, the more TI-Tregs, the worse the patient's prognosis.
Why? What are they doing in the tumor?
They're actively shutting down the body's attempt to kill the cancer cells. They create this immunosuppressive shield around the tumor, and they do it in multiple ways. First, they pump out those calming chemicals we mentioned, like IL-10 and TGF-beta, flooding the zone with stop signals.
Okay, chemical warfare. What else?
Second, they are covered in inhibitory receptors, the molecular brakes.
The checkpoint molecules, like PD-1 and CTLA-4.
Exactly those. TI-Tregs express high levels of CTLA-4, PD-1, LAG3, and others. They use these to directly engage with and turn off the cancer-killing T cells that manage to get into the tumor.
Direct suppression. And the sources mentioned a metabolic attack, too. That sounded particularly nasty.
Oh, yeah, that's the third major mechanism. It's quite sophisticated. TI-Tregs express enzymes on their surface, CD39 and CD73. What these enzymes do is take ATP, which is normally an energy currency, but outside the cell acts as a danger signal that activates immune cells, and they break it down.
Break it down into what?
Into adenosine. And adenosine is highly immunosuppressive. It binds to receptors on the killer T cells and basically puts them to sleep, blocks their activation, stops them from multiplying. It's like a chemical knockout gas protecting the tumor.
Wow. Okay, so if Tregs are protecting the tumor, the therapeutic goal is obvious. Get rid of them, deplete the TI-Tregs.
Right. But the massive challenge is selectivity. You want to eliminate the Tregs inside the tumor without wiping out the essential Tregs everywhere else in the body. Because if you do that, you risk triggering widespread, severe autoimmunity. It's a huge balancing act.
So how are researchers trying to target just the tumor Tregs?
They're looking for markers that are much more abundant on TI-Tregs compared to Tregs elsewhere. One promising target right now is a chemokine receptor called CCR8. The idea is to develop antibodies or drugs that specifically hit CCR8-positive cells, which should preferentially kill the Tregs in the tumor.
And what about other approaches, like using technology?
Yeah, nanotechnology is being explored too, designing nanoparticles that can preferentially accumulate in the tumor environment and deliver drugs, maybe something like imatinib, which can inhibit Treg function directly to the TI-Tregs, trying to contain the effect locally.
Even older tools are being looked at, like certain chemo drugs.
That's right. Low doses of some chemotherapy agents, like cyclophosphamide, CTX, seem to preferentially kill cells that are actively dividing. Since Tregs in the tumor are often proliferating more than Tregs elsewhere, low-dose CTX might selectively reduce their numbers. But again, it's less specific and carries risks.
It really highlights the double-edged sword nature of these cells.
It absolutely does. And if we kind of zoom out for you, the listener trying to grasp the big picture, this whole field is a testament to how science progresses. The work of Sakaguchi, Brunkow, and Ramsdell fundamentally shifted our understanding. It showed immunity isn't just about attack, it's about active regulation and control. Inflammation isn't just happening, it's being managed or mismanaged.
And that understanding that immunity is actively regulated opens the door to controlling it, right? Boosting Tregs for autoimmunity or suppressing them for cancer. It's guiding so much cutting-edge medicine now.
Precisely. And, one final thought, especially for anyone really digging into this topic and maybe considering a career, the sources we looked at really highlight something important about the skills needed. Studying T-cell regulation today requires way more than just knowing biology and chemistry.
Oh, like what? What stands out?
Well, for instance, a surprising reliance on physics, specifically optics. Think about a core technique like flow cytometry or FACS analysis. That's how we count, identify, sort different immune cells based on the markers they express.
Okay.
That entire technology is built on lasers, lenses, and how light scatters and shifts wavelength when it hits cells tagged with fluorescent antibodies. To really master that technique, troubleshoot it, get the best data, you actually need a decent grasp of the underlying physics principles.
Huh. I never would have directly connected laser physics with immunology research like that.
Right. And it doesn't stop there. These immune systems are incredibly complex, generating vast amounts of data, especially with modern omics techniques. So increasingly, a solid foundation in computer science and coding is becoming essential. Being able to handle large data sets, run statistical analyses, often using languages like R programming for bioinformatics, it's crucial for making sense of it all.
So the future of understanding life's defenses really lies at this intersection of biology, physics, and computer science. It's definitely something to think about. Thank you for joining us on this deep dive into the immune system's crucial regulators. We'll catch you next time.